You know, when scientists sequence a patient's tumor, what they get back is just this blizzard of data. We're talking thousands of genetic mutations. But hidden somewhere in that storm are the one or two key changes that could unlock a life-saving treatment. So how in the world do they find them? Well, today, we're going to follow the exact, rigorous journey every single one of those mutations has to take before it can ever be used to guide a patient's care.
Okay, so let's put ourselves in the shoes of a scientist. A brand new, never-before-seen variant pops up on your screen from a patient's tumor. The pressure's on. Now what? Before you can even think about a potential treatment, there are two absolutely fundamental questions you have to answer. And here's the thing. You have to answer them in the right order. If you get them mixed up, you risk making a really critical mistake.
And here they are, the two big questions. First, is this mutation biologically relevant? In other words, is it actually driving the cancer, making it grow? Is it the cause of the problem? And only after you've confirmed that can you ask the second question. Is it clinically useful? Which basically means, is there anything we can do about it? Is there a therapy we can use to target it? For a long time, these two ideas were kind of jumbled together, but modern oncology insists that you tackle them one at a time. And that separation is so important. It's what stops us from treating patients based on harmless genetic noise.
So that brings us to step one, the test for oncogenicity. Before we can even whisper the word drug, we have to figure out the mutation's basic role in the cancer. Is it in the driver's seat, flooring the gas pedal, or is it just a passenger kind of chilling in the back seat, just along for the ride? That is the absolute first question we have to answer.
So, what is this word, oncogenicity? It's simply the potential of a genetic change to actually drive a tumor's growth. And that driver versus passenger analogy is honestly perfect. The driver mutations are the real culprits. They're the engine of the cancer. Passenger mutations? They're just harmless changes that happen by chance as the tumor cells divide. This first step is all about telling them apart.
And this is not just guesswork. Scientists follow a really strict, standardized playbook. It's a scoring system that was published back in 2022 by a huge joint effort between the world's leading cancer genomics groups. And why does that matter? It means a lab in Boston and a lab in Berlin are evaluating a mutation in the exact same way. It brings this crucial consistency to the whole field.
So how does this framework actually work in practice? Well, think of it like a detective building a case. A scientist gathers all these different lines of evidence about the mutation, and each piece of evidence adds points to a total score. They are literally creating an oncogenicity scorecard for that specific variant.
But here's the deal. Not all evidence is created equal. Some clues are way more powerful than others. A slam-dunk lab experiment showing the mutation causes cells to go haywire, that's very strong evidence, and it bags a massive 8 points. Other clues might only be worth 4, or 2, or even just 1 point. And get this, there's even evidence that can subtract points if it suggests the variant is totally harmless.
Let's look at some real examples. Let's say the mutation is found in what's called a cancer hotspot, a part of a gene where we know driver mutations love to pop up. That's strong evidence, good for 4 points. What if we check huge databases of healthy people and this mutation is never seen? That's moderate evidence, and it adds 2 points. A simple computer prediction that says it might be damaging? That's just supporting evidence, only worth 1 point. You see how the case builds?
Okay, so you've gathered all your evidence, you've added up the points, now what? Well, the final score slots the mutation into one of these categories. If you get 10 or more points, boom, it's classified as oncogenic. You're confident it's a driver. But if the score is, say, between 0 and 5, it lands in this dreaded category called VUS, a variant of uncertain significance. And a negative score, that tells you it's almost certainly a harmless passenger.
Now this is maybe the most important takeaway from step 1. It acts as a super powerful filter. Only the variants that are confidently labeled oncogenic or likely oncogenic get to move on. Everything else, the VUS, the benign variants, their journey stops right here. This is an absolutely critical safety check. It prevents doctors from chasing down dead ends or, even worse, prescribing powerful drugs based on a totally harmless mutation.
All right, so for a mutation that made it past that first tough checkpoint, we can finally move on to step 2, actionability. This is where we ask that clinical question. Great, it's a driver, but can we do anything about it? And this is governed by a whole different framework, one that organizes variants into different tiers of clinical usefulness.
The system is pretty straightforward. It just organizes the variants into 4 tiers, and it's all based on how strong the evidence is for actually using them to make a treatment decision. Tier I is the best you can get. It means strong clinical significance, all the way down to Tier 4 for those variants we know are just benign.
So what do these tiers actually mean for a patient? Well, Tier I is the gold standard. It means there is an FDA-approved drug specifically for patients who have this exact mutation in this exact type of cancer. It's a green light. Tier 2 is more promising, but not yet a sure thing. Maybe a drug is approved, but for a different cancer. Or maybe there's a really promising clinical trial. Tiers 3 and 4 for now mean there's no known clinical action to take.
And this is a point you absolutely have to understand. A variant's actionability depends entirely on the context, on the specific cancer type. For example, the famous BRAF V600E mutation is Tier 1 in melanoma. We have great drugs for it. But that exact same mutation found in a different cancer might only be Tier 2 because the evidence just isn't there yet. So the final report always has to be specific to the patient's actual tumor.
Okay, so we've looked at the 2 key steps in isolation. Now, let's zoom out and put it all together. Let's follow a single mutation's complete journey, from just a line of code and some raw data to a final actionable report on a doctor's desk.
This is the entire pipeline in a nutshell. First, a raw variant is detected in the sequencing data. Then it gets put through that tough oncogenicity scoring system if, and only if, it passes as oncogenic or likely oncogenic. It then moves on to the actionability tiering system. The final results from both of these steps are then combined into a single, clear report for the physician.
Let's make this totally real with a classic example. Say an EGFR L858R mutation is found in a lung cancer patient. In step 1, it easily racks up enough points to be called oncogenic. It's a known driver. Then it moves to step 2, and because there are several FDA-approved drugs for this exact situation, it gets classified as Tier AI. The final report tells the oncologist everything they need to know. This is a driver mutation, and we have a highly effective, approved therapy ready to go.
So, this two-step process brings an incredible amount of clarity and standardization to a really complex field. But, science never stands still. In fact, there's already a proposed update for 2025 that might merge these 2 steps into one unified system. And that leaves us with a final big question to think about. As our understanding of biology gets deeper and our list of therapies gets longer, how are we going to keep redefining what makes a genetic mutation truly actionable?